Welcome our friend from Kerala, Dr. Arun Kumar TK. Dr. Arun, first of all, thank you very much for agreeing to give this presentation, and nice to see you on this platform. And
Nice
it's
to see you,
nice.
sir.
Yeah, it's the first time I'm seeing you. I mean, thank you very much. I'll request Dr. Romana Mirde to introduce Dr. Arun Kumar TK to today's audience. Dr. Romana, please.
Okay, thank you, sir. Dr. Arun Kumar TK is an assistant professor at the Department of Botany at the Zamorin's Guruvayurappan College, Kerala, and has been into mycological research for more than 2 decades. His area of specialization is fungal evolutionary biology and systematics, and has discovered and described more than 40 fungal species new to science. He has published several research articles, book chapters with international publishers, and has authored a book, The Polyporales of Kerala. He has editorial responsibility of several mycology journals. He is a curator of the AFTOL Structural and Biochemical Database, an online database maintained by the Tree of Life Consortium with collaboration across the globe. He is working on the diversity and phylogeny of mushrooms of Kerala. With this brief introduction, I welcome you, sir. It's over to you now.
Thank you, Romana. Thank you for the nice introduction.
Thank you, sir.
And good afternoon, all. It is a pleasure to be part of this wonderful group called MycoIndia. So thank you. Thank you so much for making me a part of this group, a group of very enthusiastic mycologists, I think. Okay, so thank you. Thank you. I think let me start my presentation.
Dr. Arun, I have a request. Just hide that notification. You have got some. You'll have definitely some beautiful slides of mushrooms. We don't want that notification to interfere.
Okay,
Please,
okay.
just hide it. Actually, you can see the here only. Actually, at the bottom of your PPT.
Oh, okay.
No. Okay, beautiful. Thank you so much. Yeah.
Right. Okay. Thank you. Thank you. I think my slides are visible now. Okay. So, as you all know, Kerala is a biodiversity-rich state, and we have a wonderful diversity of mushrooms over here. So I just give a brief overview of the mushroom documentation that has occurred during the past several decades in Kerala. So during the short period of time, I will just try to give you a brief overview of the mushroom diversity that we have. We all know that the most striking feature of Earth is life, and the striking feature of life is its diversity. Diversity is very important in every sense for us because biodiversity is actually wealth. Every nation has its own share of biodiversity, and we as a nation are very rich in these resources. So we know that preparing biodiversity inventories is of extreme importance because those are actually the indicators of our biowealth. Why are these biodiversity inventories important? We see that it is very important for ecosystem management and also for government policy decisions. Government policy decisions are very much influenced by the biodiversity inventories that are available. So environmental issues can be easily tackled or responsibly tackled if we have a good biodiversity inventory for each state in India. And the biodiversity, the knowledge of the diversity is very important for bioprospecting as well. As we know, we are all into bioprospecting nowadays. So in order to prospect our biowealth, we have to have a very good inventory. We have to document our resources very clearly. So these discoveries, documentations can affect the quality of human life. We are mostly concentrating on the macromycetes of Kerala. So macromycetes, there is a general term, we call it as the mushrooms. So a general definition for mushrooms is that it's the fleshy, spore-bearing, macroscopic fruiting bodies of either ascomycetes or basidiomycetes. So typically we see that mushrooms are mostly basidiomycetes, but there are also many ascomycetes fungi which produce macroscopic fruiting structures like morchellas, like the tubers, the truffles. They're all, in a loose sense, they're all mushrooms. So both basidiomycoton and ascomycoton members do produce macroscopic fruit bodies, which we tend to call mushrooms. So they are typically produced overground or on it's the nutrient source or on the substratum. So there are many terminologies connected with the mushrooms. That's one is agaric, the morels, the puffballs, the toadstools, stinkhorns, the bolides, the earth stars, bird's nest fungi. So all these refer to the different types of fruiting structures or basidiomata or ascomata that are produced, that are visible to the naked eyes. Okay, so I think most mycologists know how a fruiting structure is formed. It's from the collection of mycelia, and mycelia aggregate to form beautiful structures above ground when it's trying to reproduce, when it's time to disseminate their spores. Okay. So in the basidiomycoton members, we usually tend to find that there are two major subclasses, Tremellomycetidae and Agaricomycetidae, that tend to have members that produce mushrooms. Of course, there are many classification systems of fungi which categorize or classify fungi differently. I have just used this classification that was by Kirk in 2001. Actually, there are several other classifications which came after that. Recently, we have the classification systems by Tedder-Su and et al. So there are many groups which have been split up and which have been combined to make different taxonomic ranks. So this one that I have adopted here is a very easy to follow one, where there are two subclasses for the basidiomycetes class, the Tremellomycetidae and the Agaricomycetidae. So in Agaricomycetidae, there are 8 major orders that have been recognized. And all these 8 orders have members that produce macroscopic fruiting structures. Those are the agaricales, the polytails, the chanterellales, the hymenocetales, phallales, polyporales, russolales, and the pelliforales. Again, there is another major subclass, that is the Tremellomycetidae, which usually tend to produce many of the jelly fungal structures, or many of the basidiomycoton yeast species are actually included in this Tremellomycetidae. So these are the basidiomycetes members which produce mushroom structures, mushroom fruiting bodies. And there are also some ascomycete members which also tend to produce macromycetes, or mushroom-shaped structure, or mushrooms. We can call it macromycetes, fruit bodies. Those are the helotales, the hypocreales. So in helotales, you will find the earth tongues, the geoglossum, the trichoglossum, etc. Then there is the hypocreales, which produce in cordyceps species. Neolecta. Neolecta is a cylindrical fruiting structure which is produced by a ascomycete member. Then there is the pessicales, which produce the morchella, the tubers, truffles, etc. Then Sileria is another ascomycoton member which produces a branched fruiting structure. And Daldinia is another ascomycoton member which also produces a macromycetes, a macroscopic fruiting structure. And we know that mushrooms have many economic importance. Its edibility, medicinal properties, then many industrially important properties, all make them very sought after. We know that Ganoderma, the preparations from Ganoderma, psilocybes are all commercially available nowadays for treatment of several ailments, for health improvement, as nutraceuticals. So edible mushroom industry, as well as the medicinal mushroom industry and nutraceutical industry, very much rely on many of these mushrooms. So mushrooms are economically important, we all know. Then there is a major ecological role that these mushrooms do play, or the fungi, the basidiomycoton and the ascomycoton members do play. That is the degradation and ultimate recycling of nutrients. So they degrade plant material effectively and recycle the nutrients which are trapped in the plant bodies. They also act as food sources for many insects, and many small rodents and small animals. So in effect, they have a very important, an extremely important environmental role. Documenting the mushrooms of Kerala. So Kerala is a state in the southwest corner of India, and we have a total area of 39,000 square kilometers. And we have a forest cover, which is around 11,000 square kilometers. We form a part of the Western Ghats. So along with the other states, we also share some part of the Western Ghat flora. So India, Kerala has got a rich biodiversity in terms of the plant wealth. We have around 5,000 plant species recorded so far, and even now some new species have been recorded. Regarding the plant species. So we have a good biodiversity. The mushroom research in Kerala actually have a 4-decade experience. It actually started with Professor K.M. Leelavati at the University of Calicut, the Department of Botany, University of Calicut, and followed by her students. Professor Manimohan, Dr. Vrinda, Dr. C.K. Pradeep, they are all students of Professor K.M. Leelavati. And after the generation, there is another generation of young mycologists coming up, still working on mushrooms of Kerala. So mainly we have 2 major groups that work on the mushroom research in Kerala. That is one on the southern part of Kerala, that is by Dr. C.K. Pradeep, who is working at the JNDBGRI, Tropical Botanical Garden and Research Institute at Trivandrum. And then we, at the northern Kerala, we have myself and Dr. Anil Raj and Dr. Deepna, who work on the mushroom diversity. So together, all these mycologists for 4 decades have summed up or reported a number of fungal species from the forest areas as well as the human inhabited areas of Kerala. We have a published report of around 1,000 species of mushrooms from Kerala. Let me repeat, 1,000 species of mushrooms alone, not fungi, mushrooms. So there are around 1,000 species reported from Kerala, of which Basidiomycoton members are around 936 species. And as we know, the mushrooms, again, the Basidiomycoton mushrooms can be again be classified as guild and non-guild, as well as a group called polypores. So there are the guild mushroom fungi, which we usually have the agaricus and such mushrooms. Then there are the non-guild ones like the boletes, then the jelly fungi. Then we have the polypores like the Ganoderma. So around 936 species of these guild, non-guild, and polypore members have been reported from Kerala so far. With regards to Ascomycota, we have 12 species that is recorded. Not all the Ascomycota members, but it's the large fruit body forming Ascomycota members. Of course, we have a number of Ascomycota members which are plant pathogens. So that is a huge list. But regarding the Ascomycota members which produce macroscopic fruit bodies, we only have 12 species reports, which is quite interesting because it is not the scant Ascomycota members, but it is because of the relatively very less studies that have been carried out on this group. So Ascomycota, the large fruit body forming Ascomycota members have been relatively untouched in Kerala. So we only have 12 species, that is 8 species of Sileria, then 1 Daldinia species, and 3 Cookiana. We have a lot more, of course. And as I said, we have a plant species diversity of around 5,000. And if we try to estimate the number of species of fungi that we will have to resort to the estimate calculations by Hawksworth and Blackwell. Of course, in Blackwell in 2001, actually predicted a 1:10.6 plant fungal species ratio, and she estimated that there should be around 10 million, 5.1 million fungal species that exist on the Earth. So if we take that 1:10.6 ratio, which is very conservative for a tropical country, still we have an estimate of almost 58 or 57,000 species, an estimate. And the recorded number of fungus species from Kerala is around 5,000. 320. The recorded, so the mushroom species that have been recorded from Kerala is about 18% of the recorded fungal species. And out of the 948 species of mushrooms that have been recorded, we can find that about 40% of them were new to science. So you have to just see how much novelty there exists in terms of species in a small geographic area or a state called Kerala. We had reports of 5 newly described genera. So there were 5 new genera that have been described and published from Kerala alone, and almost 40% of the fungi that are being reported from Kerala is new to science. So we can just imagine the number of mushroom species that must be existing outside the state of Kerala, all around India. There are many areas in India which are underexplored, and we should be really concentrating on exploring the mushroom diversity of other parts of India too. So India is a large area with a 32 lakh square kilometer area, 3 lakh square kilometer area. And even from the 39,000 square kilometer area, we have a diversity of around 1,000 species of mushrooms alone. Okay. So when we speak about mushroom diversity, we know that the documentation is extremely important. New taxa are still being reported every month or so, and we have to have a thorough knowledge of the extent of diversity we have. So this mushroom diversity actually poses opportunities as well as challenges for us. So let's briefly see what are the opportunities. So the diversity richness, actually, the discovery of unexpected leads to unexpected variety of products that can include chemicals, genes, metabolic pathways, materials and behaviors. So these all can provide physical blueprints or inspirations for new ideas. So if we tap the diversity of mushrooms we have, it's a huge mine. And there is a vast ethnomycological data that we have amongst us. Even in Kerala, there is a large ethnomycological knowledge that exists mostly with the tribal areas. People have a lot of experience and knowledge about using wild mushrooms as medicines and as edibles. So cataloging, documenting, publishing all these is extremely important. There are many underutilized wild species which can be which might be having several traits which can be commercially utilized. Then there may be several species which can be used for the protection of several industrially important compounds, antibiotics, novel compounds, etc. So those are the opportunities that we have. Of course, we also have some challenges that we face because documentation and diversity is still at a nascent stage in many parts of India. There are many misidentifications that actually happen, and this is one area. Misidentification is one area we all should be concerned about, because identifying a taxa accurately is very important in order to utilize it from a biotechnological perspective, from an applied perspective. We have to have a perfect, correct, accurate identity for a species. Otherwise, there can be all sorts of mixes, and we might end up with not so dependable results. So there are several underutilized germplasm available. Then habitat destruction is another area which we in Kerala face, because we have a small land area with a very small forest cover now, and even that forest cover is fast deteriorating. So we are losing most of our forests, and along with our forests, we are losing most of the fungi. So we should be at a war footage of not only documenting, but also conserving these mushroom resources. We tried as much as possible to make cultures of these and try to maintain. That is one of the methods that we can do a little bit to save all these species. Okay, so long-term resource management strategies are extremely important. It's not that one or two people work in these areas, but there should be concerted efforts and even support from the government organizations. Okay. So let me move on to the taxa that have been documented from Kerala, the mushroom species. So we found that there is the Agaricomycetidae and the Tremolomycetidae. In Agaricomycetidae, we have several orders. the agaricales is the most dominant order of all mushroom forming fungi in Kerala. In a worldwide perspective, there are around 13,000 described species worldwide, and most of these agaricale members are dominated by pileate, stipitate forms with a gill or a lamellate hymenophore. Of course, there are exceptions, and almost 700 species of agaricale members have been reported from Kerala. Some of the very closely studied or thoroughly studied groups are the Plutiaceae group, which is studied by Professor Dr. C.K. Pradeep. Then there are the dark spot agarics, which were studied by Gracious Thomas and Professor Manimohan from Calicut University. Then there is the Lepiotaceous fungi, which I studied personally. Lepiotaceous fungi is a litter decomposing group that includes Lepiota, Leucogaricus, Leucocoprinus. Then there is the Amanita group, which has also been fairly studied. Entelloma is a group which was studied by Dr. Anil Raj. And Inocypes is another group studied by Deepna Lada, and again, a student of Professor Manimohan. Then we have the Hygrocypes and the Mycenas from Kerala. So 2 books have been published already on the agaricales, that is on the Mycenas of Kerala and from the Inocypes of Kerala by Professor Manimohan and his students. Okay. So let me move on to the photographs of some of the agaricales members that we have recorded. I cannot include all the photographs that I have, of course, because there is a time crunch and we have to, I think I should strictly follow the time schedule. So this is the Agaricus, a trisulfuratus species. There is an Agaricus species that we have in Kerala. Then there is another Agaricus species. We have the Clarkindia trichotis. This is a Clarkindia species which has been collected. This species has been collected from Kerala and from Sri Lanka very commonly. This is another Leucocoprinus species that is always collected from dung, cow dungs. And Termitomyces species, which is quite common all over Kerala. This is Termitomyces microcarpus species. We have many other Termitomyces species. Around 13 species of Termitomyces are present in Kerala. And then we have the Microsaliota, which has got an association with the ants. Ant colonies always tend to associate, or these Microsaliota always has been observed to fruit from the ant colonies or near the ant nests. Then we have the Parasolas, we have the Volvariellas, the Volvariella mushrooms, which usually produce on decaying organic. The Lepista. We have the Trichomopsis crocobuffa, Trichomopsis species. Then we have the Hygrocybe. There is the Mycena. This one is actually bioluminescent. This is a bioluminescent fungi, which is Mycena. Then we have the Marasmius species. This Marasmius hematocephala. Then we have the Omphallotus oleireus, which is again another bioluminescent fungus. Coming to boletails, so there is another order called the boletails, which with around 1,400 described species worldwide. This group is dominated by the pileate, stipitate forms with a tubular or sometimes lamellate hymenophore. The most of the major genera of boletails that have been documented from Kerala is Boletus, Paxillus, Philibopus, and Buchwalder boletus. So this is the Strobidomyces species that have been collected from Kerala. There's Scleroderma nitidum. This is Philibopus species, Philibopus portland. This is actually an edible species and which is commercially cultivated in China and Thailand. I personally do not know if there are any cultivators who commercially cultivate this in India. But this is the wild edible species and much sought after one. This is Parvis rocomus species. This is a Boletellus species, which we collected from Kerala. Then we come to the Chanterelles group. Of course, Chanterelles is a small order with around 200 or so described species worldwide. And Filaria, Cantharellus, Clavulina are some of the species that we have in Cantharellus. Cantharellus has a wide range of fruit body shapes, ranging from clavarioid or a club shaped mushrooms to almost pileate or spatulate mushroom-shaped fruit bodies. Cantharellus is an example where you will find a pileate or spatulate mushroom-shaped fruit bodies present. And Afilaria is one where you have a clavarioid, a coral-like fruit body structure. Then there is the clavulina, which again comes in the chanterelles. It is again a coraloid group. Then we have the clavulina humilis, again, which is a Catharella group. And then we have the hymenocetales members with around 900 species described worldwide. They are the steroid, corticoid, hydnoid, basidiomata forming fungi. The examples are Rigidiporus, Felinus, Inonotus. So from Kerala, we have around 32 records of these hymenocetales members. Hydnoporia is a member of the hymenocetales. Then we have the hymenocete, hymenocete Bordingi, then the hymenocete acromarginata. These are the hymenocetaceae members, hymenocetales, the Coltrecia, the Philoporia. Then coming to polyporales group, we have the then we have the phallales group, which has got around 100 described species, and Clathrus and Phallus is the most dominant genera in Kerala. This is a Dictyophora, and which has now been placed under Phallus. Earlier it was a Dictyophora, now it is Phallus intuslatus. Okay. Then we have the gomphales. The gomphales members again produce spatulate shaped or else coral shaped fruit bodies. Ramaria pusilla, then another species of Ramaria. Several Ramaria species have been reported from Kerala. So these are all the Ramaria species that we have, which belongs to the gomphales. Of course, we also have a gomphus, a new species of gomphus, which is with a journal and will be published soon. Then we have the polyporales, the one of the largest orders of the wood rotting fungi, with around 1,800 described species worldwide. The polyporales members of poroid, hydnoid, lamellate basidiomata, and we have 92 species that have been recorded from Kerala. The major genera are the Ganoderma, lentinus, Panus, Polyporus, Rigidoporus, and Tremetus. Of course, we have published a book on the polyporales of Kerala. Another book on the hymenocetales is currently under publication. So these are some of the polyporales members: the Spongiporus, the Flavodon, Irpex lacteus, Nigroporus, Flebia ludoviciana, then Rigidoporus lineatus. We have the Terrana. This has got a wonderful cobalt blue coloration. Then Jercandra, Adesta, Oxychete, Panus, several species of Panus, Panus similis, Panus neostrigosus, Panus velutinus, then several species of Podoscyphas, Podoscypha venustilla, Podoscypha petaloides, Podoscypha involuta. Then we have many species of Lentinus, Lentinus arcularius, Lentinus dicolamelatus, Lentinus quorosulus. There are so many species of Lentinus which have been reported from Kerala. Then comes the Ganoderma species. We have around 11 species of Ganoderma from Kerala alone. And as we know, Ganoderma taxonomy is very complicated, and there are several misidentifications that actually happen with Ganoderma. Many of the fungal pathogenic Ganoderma species have been misidentified for even about a century. Some of these species have been misidentified. And recently, morphology as well as combined morphology as well as morphological analysis is helping us to clear off some of the confusions regarding the Ganoderma identification. So this is Ganoderma pseudoacronatum, which was a newly described species of Kerala. Then we have the multipilium. We have the Ganoderma kerulans, which is actually associated with the stem rot disease of coconut trees in Kerala. Then we have the Ganoderma tropicum, Ganoderma multiplicatum. Then we have the Sanguinoderma, which is a pileate polypore species. All these are wood rotting fungi. And then there is a Lignosus or rhinoceros, which is known as the tiger milk mushroom. It's got a high medicinal property, and this is actually, I think, commercially cultivated in Thailand. But this was the first report of this rare medicinal mushroom from Kerala, and this particular one has a high scope of being commercially cultivated, because it can be grown in culture as well as can be utilized for its medicinal properties. And then we have the microporous Violaceae cinerescens, then the pseudofavolus tenuis, then we have the microporous xanthopus, pycnoporus, another species, Cinnabarinus, polyporous dictyopus, cellulariella, Earliella, Leotremitus, Tremitus, several species of Tremitus, Tremitus mariana, Tremitus maini. Of course, this green coloration is due to the growth of some algae over the hirsute spinal surface of these mushrooms. Then Dadelia. So these are some of the polyporales members that we have. Then we have the Rasulales documentation. Of course, Rasula is one of the mushrooms that have been very frequently encountered. A fair number of documentations have been done, but still there are many species that have to be documented. So a concerted study of Rasula is actually required from care. This is a Rasula species. Then there is another major mushroom forming fungi, which is the jelly fungi, or fungi that produce macroscopic fruiting structures. So it actually belongs to the Tremellomycetidae subclass, where we have several orders. Auriculariales, the tremellales, and the Dacrymycetales are the dominant jelly, the macro fruit body forming jelly fungi. So we have some of the members. This is the Tremella species, another Tremella which was newly described from Kerala, Tremella poicavensis. This is a Cirrobasidium species. This is a Phaeotremella. This is a Caloceros species. This is Auricularia. This is Dacropinax. And another interesting fungi, which actually does not belong to the Agaricomycetidae and the Tremellomycetidae, is Auriculoscipa. This is actually a rust member. This is a septobasidial member, and it actually a rust, a uridinales, belongs to uridinales group, but still produces a macroscopic fruiting structure, and it is an example of a fungus insect plant relationship. So there is a coccid or a scale insect which actually feeds on anacardiaceous trees. There's cashew and mango here, and these coccids or the scale insects are in turn parasitized by these Auriculoscipa. So it's actually these are insect parasites which feed, and they usually tend to support the colonies of several insects inside them. It's like kind of a septobasidium-like relationship, a kin relationship, where they can feed on some of the insects while the other insects are being protected. So this is a fungus which has been documented from Kerala, but not yet documented from any other parts of the world. Okay, so and these are my students, and they are actively involved with documenting mushrooms. Vinjita was involved with documenting the polypore diversity. She was awarded a PhD recently. Then Krishna Priya worked on the clavarioid fungi of India. She also was awarded a PhD recently. Both of them documented the polypores and the clavarioid fungi. Now Anjita is working on jelly fungi of Kerala. Salna is working on the polytoid fungi. The Gina is working on, again, the molecular aspects, molecular phylogeny of Lepiotaceous fungi. So this is my lab group, and together we are joining to document the diversity of mushrooms here. Okay. So I think I stuck on to time. It took a little bit more. Sorry.
Oh, that's okay. That's okay, Dr. Arun. Thank you. That was a beautiful work with the beautiful slides. Thank you very much. And may I ask the first question to you, please?
Oh.
How challenging is the culturing of mushroom species, actually?
The thing is, some of the groups of mushrooms are very easy for cultivation, but some of the groups are very, very difficult. Especially, we found that the polyporales members, many of the polyporales members have been difficult because of contamination issues. And because since the polypore fruit bodies may be perennial, most of the actively growing mycelium may be absent. So we found it very difficult to isolate the polyporales members. as well as some of the agaricales members. The jelly fungi group is a really relatively easy one to deal with. We have been partially successful in culturing some our collections, but not all collections were yielded to culturing.
Do we have a very dedicated culture collection for mushroom cultures?
We have some dedicated in Solon, but I think one dedicated to mushrooms is extremely needed. Is
Yes.
actually needed, yeah, yes.
Thank you so much. Yeah, I will let other people ask. Dr. Sunil Srivastava, please.
Yes, thank you, Dr. Arun. It was a brilliant presentation and such a vast biodiversity of the mushrooms, which I'm not aware of. So Kerala is a gold mine, of course, for many plants, and it's very enriching experience to know about all this. Kerala also happens to be one of the Ayurveda centers, the tribal medicine and all that. So, do you find any kind of a mushroom documented which is being extensively used for some unique medicinal properties which we are not aware in the northern part?
Yes.
Because
Actually, in the Ayurvedic texts, I have not come across any mention, but there is a vast ethnomycological knowledge that we have. So when we go for collections, we come across several tribal people who actually use them as medicines, and most of these mushrooms have been used as medicines by them. And some of those have been documented by Professor C.K. Pradeep of TBGRI. So some of those are available, but still a lot has to be documented. And most of these are with are very protected knowledge, actually.
Yeah, but we should look into it because these are really gold mine.
Yes, yes.
Thank you.
Dr. Sudhir, please.
Yeah, Arunji, I have one question. Have you ever got success for culturing termitomyces species, or do you
No.
have some?
So no, I have tried it, but I have not. Maybe I don't know whether anybody has succeeded so far with termitomyces. I'm not aware, but till now.
What could be the reason behind it? Just because it passes through gut of these termites or something like that, or something?
Yeah, maybe. I think that may be a reason because they have been associated with the ants for long, and I think some of those chemicals that the ants secrete may be required for fruiting. Or I'm not actually. Yeah, I was not successful in cultivating termitomyces.
Yeah. Okay. Thank you. Thank you.
Ms. Nita Fatima, please.
Thank you, Arun, for the wonderful talk. It was really lovely to see your presentation. I want to ask if, while identifying the mushrooms, did you use morphological characters alone, or did you use molecular tools as well?
We used both. We use the combined approach where we use both morphology and molecular approaches, because with morphology alone, we cannot really pinpoint mushrooms to species level. It's very difficult because mushrooms have very little or taxonomically important characters. So there can be several misidentifications if we go with morphology alone, but that is not to say that we have to rely on molecular methodology alone. Both has to be combined. So both morphology and molecular techniques were used.
Also, do you know of other resources for mushrooms for a repository of their whatever we have in India, across the country or something?
You mean culture collections?
Not culture collections, like books detailing the mushrooms found in India, like a field guide or something.
There are several books available regarding the identification of mushrooms in Kerala. For Russells, we have one by Kannada's. Then for some of the groups in Kerala, we have for the inosides, the mycenas. Then for the polyporales, we have one, the polyporales of Kerala. So there are several books actually available for identification, but not for all groups, but for selected few, because the diversity is so vast that we cannot combine everything into one publication. So yes, definitely there are available.
Thank you, sir.
The last question would be from Mr. Hadil Haq. If I'm not pronouncing it wrongly, the name. Mr. Abdul Haq.
Senior sir, can I ask a questions?
Yes, please, Dr. Singh, please.
Ian Singh, good afternoon, Arun sir.
Good afternoon, sir.
As you discussed earlier that the termitomyces, someone is asked that questions whether it is culturable or not.
Yeah.
So recently, though I am a worker of this micromycetes, means lower fungi, but I am trying to culture some higher mushrooms also. So recently, one species of this Agaricus agaricari, it is sent for this publication by phylogenetic study in that fungal diversity. But recently, 2 weeks ago, I isolated one species of this termitomyces, and by molecular analysis, it is saying that termitomyces hemi, okay, and it's a pure white fruit body. So while collecting, I was not imagining that it can be a termito. I was before this by seeing in that nature by naked eye. So I was not hoping that it could be a termitomyces, but once I dig out that, so that a long stripe and other thing, that conic structures was found, and luckily it was isolated. So 1 year, 2 years back, even 3 species, that one bigger species I have isolated, and still it is under analysis, means molecular identification. But hemi, it is already clear that it's a termitomyces, and phylogenetically it is a hemi species. And so 3 I have isolated luckily. So I don't know, means though it was associated in it was growing on this pasture land, and one was in my Agharkar Research Institute campus. So 3, that 2 is the bigger and 1 is the very small fruit body. So okay, another query, this recently, because this is a general point, means in Australia recently 3 people have died. One perhaps that relative, she cooked and they have eaten, and 3 have died. So whether it is intentionally, I don't know that what was the genus and what is the region. Knowingly they have eaten or knowingly they have prepared to kill them. So it is in news recently, 2 weeks back in Australia. So that I don't know, but it's very
Yeah.
curious. Yeah.
Actually, misidentification is a major reason for this mushroom poisoning. Many people misidentified because most mushrooms may look alike. Yeah. So misidentification is actually a problem.
Yeah. So thank you very much, sir. It was a nice presentation of this related to these mushrooms, nice pictures and that
Thank you.
morphology of that mushrooms. So thanks a lot. It was a nice presentation. Yeah.